Good afternoon. The categories of my study are chemistry and English. Title of the study is The Influence of Sugar Concentration on Microorganism Growth, as you can see on the screen. This work was completed by 11th grade student Колесова Марія Dmиtrівна. Objective of the study: to determine how varying concentrations of sugar affect the growth and reproduction of microorganisms in chicken broth. Relevance of the study: sugar is commonly used in food production and preservation, while moderate amounts can serve as a nutrient source for microorganisms. Excessive sugar may inhibit their development. This study helps better understand how sugar affects microbial environments, which is crucial in food safety, microbiology, and health sciences. Moreover, since similar processes occur in human body, understanding the influence of sugar on microbial growth highlights the importance of limiting sugar consumption for overall health. My hypothesis sounds so. Moderate amounts of sugar will promote the growth of microorganisms, whereas high concentrations may inhibit their reproduction. Research tasks sound so. Firstly, we should observe and document the development of microorganisms in different sugar environments. Then we should compare the rate and density of microbial growth depending on sugar concentrations. Then to analyze the data to identify patterns in microorganism response to sugar. Then we should draw conclusions about the relationship between sugar concentration and microorganism growth. In the end, it is better to explore the chemical role of sugar in microbial metabolism. Methods of the study: I used observation, comparison, measurement, data analysis, and chemical analysis. Materials that were used: 3 sterile petri dishes, chicken broth, granulated sugar, measuring spoon, and natural exposure to air as a source of microorganisms. Procedure: prepare 3 petri dishes with equal amounts of chicken broth in each one. Leave the first dish with no sugar. Add half a teaspoon of sugar to the second dish and add a full teaspoon of sugar to the third dish. Leave all 3 dishes uncovered for a short time to allow airborne microorganisms to settle. Cover the dishes and place them in the same warm environment. In the end, observe daily over several days or weeks. Chemical background of my experiment. Sugar serves as a carbon source for many microorganisms. During microbial metabolism, sucrose is broken down into glucose and fructose, which enter the glycolysis pathway, the first stage of cellular respiration. Through this process, energy in the form of ATP is produced, which microorganisms use for growth, reproduction, and maintenance. Hydrolysis of sucrose is seen on the screen. In high concentrations, sugar creates a hypertonic environment, which in some cases may inhibit microbial growth by drawing water out of microbial cells. However, many molds and yeasts have adapted to thrive in sugary environments, especially when there is moisture available, as in our broth. That was actually the reason we chose it. Therefore, the presence of sugar enhances microbial activity up to a certain point, supporting rapid proliferation by fueling cellular respiration and biomass synthesis. Here you can see my experiment. The reference is in 2 weeks. In the third dish, with 1 teaspoon of sugar, microorganisms appeared first and showed the most rapid initial growth. In the second dish, with half a teaspoon of sugar, growth appeared shortly after and was moderate. In the first dish, with no sugar, mold appeared last and spread the slowest. Analysis of results. The data indicate that a high sugar concentration, which is 1 teaspoon, promotes the fastest and most active initial growth of microorganisms. The moderate sugar environment, half a teaspoon, supported microbial development at a steady pace, while the absence of sugar resulted in the slowest onset and spread of mold. This suggests that sugar concentration directly influences microbial growth rate, with high concentrations offering more nutrients for rapid reproduction. Chemically, this confirms that sugar provides the carbon and energy necessary for microbial metabolic pathways. More sugar means more fuel for growth up to a threshold. Conclusion. A high sugar concentration supports the rapid reproduction of microorganisms. Moderate sugar also promotes growth, but to a less extent. No sugar results in the slowest microbial development. Sugar serves as a key nutrient in microbial reproduction, and its concentration determines the speed and extent of the full growth. From a chemical perspective, sugar fuels vital biochemical pathways in microorganisms. Since sugar similarly impacts microbial behavior in the human body, excessive consumption may alter natural microbial balance and health. Therefore, it is really recommended that people reduce their sugar intake to maintain healthy biological processes and prevent negative health effects. Here you can see my scientific sources, and I'm really thankful for your attention.